So I want to again remind you of our big picture, which is that we're talking about the fate of chemical reactions. We've finished discussing direction, but the other key thing is the rate of reaction. So even if a reaction is moving in a spontaneous direction, it can be slow or fast depending on the important factors we're going to talk about now. So in living cells, the rate of reactions are controlled by enzymes. Enzymes are protein catalysts. Catalyst means something that speeds up a reaction. Enzymes are made of proteins. So remember, that means they're made of amino acids. So a spontaneous reaction is not necessarily a fast reaction. I mentioned this earlier. It might take 1,000 years for that spontaneous reaction to occur. Well, the cell doesn't have 1,000 years to sit around and wait for a chemical reaction to happen. The cell's not even going to be alive in 48 hours, let alone 1,000 years. So cells need reactions to happen faster than they otherwise would, and that's why they rely on these enzymes. Okay, so enzymes are not going to change the direction of the reaction. So they do not change the direction. They just make whatever direction the reaction is going go faster. Okay, that's all that an enzyme can do. So to explore how enzymes work, we need to talk about why reactions would be slow. So if a reaction is favored by thermodynamics, why wouldn't it happen fast? The reason is that every reaction requires a different type of energy. I know you're sighing right now thinking, oh my God, all these forms of energy. But it's just the way it is. So activation energy is going to activate the reaction. Okay, so if that helps you remember, it's going to activate the reaction. So it's this small amount of energy to get the whole process started. It's going to get molecules close together because, of course, you can't have a reaction. If two molecules are on the other side of the room, they can't react with each other. So part of this is just getting molecules near each other. Once they're near each other, they're going to be able to go into sort of a transition state or a state in which they're ready to react with each other. And to get all of that accomplished, we have to have energy. Even if it's an exergonic reaction, which we said earlier is going to release energy eventually, I'm still going to have this little bit of input. So the most typical way that this is achieved is by heat. So this is why reactions will be sped up if they're warmer. And if you know, molecules move faster when you heat things up. So if I have two molecules far apart from each other, if I start heating them up, they move faster and they'll get close together faster. So that's the key way that heat will provide activation energy. Now again, for the cell, it's not practical to have high heat to speed up chemical reactions. Remember, our bodies have to be at a very specific temperature. They can't be hotter than that temperature or it's bad for us. So our reactions need to happen at our body temperature. And if you think about other organisms that don't have our body temperature, like that are cooler in temperature, like a bacteria that lives in the soil, for example, they have to have their reactions happen at even lower temperatures. So the cells are not going to be able to rely on heat alone for activation energy. So the strategy of living things is to have enzymes. And what enzymes do is lower the activation energy that's needed. They don't eliminate it entirely. You still have to have some, but it's much lower. So this means that less heat is needed to activate the reaction. So now just a small amount of heat, whatever is available in the cell, or at the body temperature of the organism, can now push the reactants into that transition state where they're ready to react. So this is often represented graphically. So on the y-axis of this graph, we have the amount of free energy. And at the beginning of the reaction, we have the G energy the free energy of the reactants. And at the end of the reaction, we have the G of the products. And so if I look at this graph, I'll just imagine some numbers here. This might be 5 because it's less, and this might be 25. So if my products have less free energy than my reactants, then I must have a negative free energy change. So this reaction must be exergonic. Right? So this is what we've called a spontaneous reaction. It will happen on its own. It will move that direction spontaneously. and it will release energy. So we can also think of it as going downhill. So the ball is rolling downhill, releasing energy. But before it can roll downhill, there's this hump. This hump, this amount of energy here, so going up in energy, is the activation energy. The red line, so this first line here, that's the activation energy without an enzyme. So I'm going to have to put in that much heat in order to get the reaction to occur. The blue line is with enzyme. So when I have an enzyme, that bump is a little bit smaller. Do I still have to have some energy? Yeah, you bet. This reaction is going to still need a little bit of heat energy, but it's much less. So this reaction will happen faster in the presence of an enzyme because it will take less time to get this much heat energy. This is the fundamental principle, too, behind putting food in the fridge, right? So a lot of our food is living cells. So we're trying to slow down those living cells carrying out reactions, like the bacteria carrying out reactions on our food. And so if we put them in the fridge, they can't get even this small amount of activation energy. So how do enzymes lower the activation energy? It depends on the enzyme, but in general, it's going to help get molecules together. So the enzyme, so I'll draw a little enzyme here, right? If it's going to help us carry out this reaction, it's going to bind A and it's going to bind B. So it's going to go around collecting A and B and putting them together. So that's going to make the reaction go faster. Once they're together like this, that can help with bonding. It can bend the reactants. It can change the microenvironment around these, which are different ways to just get them ready to react, to go into that transition state where they're ready to react with less heat. So enzymes are proteins, and so remembering back to chapter 3, because they're proteins, they have very specific shapes. That specific three-dimensional shape will only match certain reactions. So every reaction in your body has a unique enzyme with its own unique shape. That shape in particular has a very important part called the active site. So I draw a little enzyme earlier where I had A and B. This space that I've drawn here would be the active site. The active site is the location in the enzyme where the reactants bind and the reaction takes place. So in this figure here from your textbook, here's my enzyme, here's my active site. The reactants, in this case glucose and ATP, will come in and they create something called the enzyme substrate complex. Once the reaction occurs, they no longer fit in the active site. Remember these matching shapes we talked about in chapter 3, and so these guys will float out because they're done. Right? So the enzyme binds the substrate, the reactants in the active site, forms the enzyme substrate complex. The reaction occurs, and then the products, ADP and glucose phosphate in this case, are released. So again, I said this earlier, but I just want to emphasize it again. Every single reaction has a unique enzyme. And this is because of the unique three-dimensional shape of every enzyme. So enzymes have very high affinity or specificity, those words kind of mean the same thing, for a particular substrate. So substrate is another word for the reactants. And it's sometimes called the lock and key fit. So you have your lock, you have your key, and if you have a match there, then it will open the door or carry out the reaction. And this is just a little image here that's it's research images showing the shape of an enzyme that was determined by the researchers. And what they noticed was if you look at the enzyme before and you look at the enzyme once it's bound to its substrate, the shape actually changes. So I'm going to circle this area for you in particular. So notice how there's a gap there, and now that gap closes. So you have your enzyme, you have your substrate. Not only does your substrate fit into the active site, and let me use my pen here just so you can see this a little better. Not only does the pen fit into the active site, but the enzyme closes around it if it's the right substrate. If it's a different pen in my little scenario, so now this is similar shape but not the right shape, notice that the enzyme doesn't close around it. Because it doesn't close around it, it just floats back out and no reaction occurs. If the right substrate comes in, so this is the matching substrate, the enzyme closes around it and the reaction occurs. That's something called induced fit. So it's such a specific matching shape that you will only get that closing with the perfect shaped substrate, and only then will reaction occur. So that's why every single chemical reaction will have a unique enzyme that carries it out with a perfect matching shape. So enzymes are very vulnerable to their environment. Most enzymes are going to have a really specific range of temperature and pH, for example, that they operate in. So this is a graph showing the rate of the chemical reaction, which is a readout of the enzyme function. So this is really telling us about enzyme function. I have the highest enzyme function at around 37 degrees C. And if you remember, that is equal to body temperature. If my temperature is too cold, okay, if my temperature is too cold, then there's no heat. So even though my enzyme could still work theoretically, there's no heat to provide that little bit of activation energy that's necessary for the reaction to occur, so I get less reaction. On the other side of the graph, something very different happens. There's plenty of heat, but I'm still getting less enzyme activity. This goes back to another concept that we talked about in Chapter 3, denaturation. High heat unravels the shape of proteins. If my shape of my enzyme is messed up, it no longer is correct, then it cannot bind the substrate. If it can't bind the substrate, then no reaction will occur. So again, let me just reiterate here. At 37 degrees, this is probably from a mammal like ourselves, which is our body temp. I'll get maximal function of the enzyme. If I get colder than that, then there's not enough heat to provide activation energy for enzyme activity. And so even though my enzyme could still work, I can't get that little bit of activation energy that I need. The enzyme is okay, it's intact, and if you think about it, as long as it heats up, then that food will be fine. And if you think about like a meat in the fridge, right, I could still take that meat out and the chemical reactions will occur and it would go bad. Denaturation, on the other hand, would be like cooking that meat. That high heat denatures that meat. And so it changes the shape of the proteins that are in the meat. And that is the enzymes that are in that meat are not okay anymore. pH has a similar, slightly different effect on enzyme activity. So if I draw that graph, I get optimal activity at what I hope you would say is, if I could talk to you right now, is that it would be pH 7, because that's our body pH. So most enzymes operate at pH 7. If I go either direction towards a more acidity or less acidity, then I will get a decrease in enzyme activity. In both cases, it's denaturation. So if I have too little acid or rather too basic or too acidic, I will denature the enzyme and the enzyme will no longer work. So the key here is denaturation and the shape of enzymes for pH, and for heat, high heat denatures, but not enough heat doesn't give me the activation energy that's still required to get over that bump and carry out my chemical reaction.